Everyone. So exciting time ahead. Big thanks to everyone joining our awesome Heredity Biosciences webinar. The goal is to spread awareness, and we promise more fun events like this in this future. So firstly, a super welcome to all of you to today's special webinar. I am Dr. Gopal K Purohit, CEO and co-founder of Heredity Biosciences. I'm really happy to have you all here with us for this fantastic webinar today. In today's webinar, we investigate into the transformative domain of medical genetics and the evolution of next-generation sequencing technologies. So NGS, rapidly sequencing genes or entire genomes, deleting mutations, crucial for genetic diagnosis, progenesis, treatment decisions, and patient monitoring. It is a faster sequencing capacity, open doors for personalized precision medicines. A heartfelt thank to all our esteemed guest speaker, Ms. Sohni Tagore, research analyst at the Genebox Mumbai, Maharashtra. Your acceptance for our invitation has enriched today's webinar, and the entire Heredity Biosciences team is truly grateful for your presence and expertise. Mr. Sohni, your valuable contributions has significantly enhanced the quality of our webinar. Thank you for your being an integral part of these enlightenment sessions. Now, audience, the highlight of today's webinar is Cantor, an intrinsic study of advancement in the next generation sequencing. So next generation sequencing by the medical genetics, the advanced next generation platform, the transplanting of the field of genomics by allowing to the partial sequence of millions, 2 billions of DNA fragments. The capacity has unlocked a new opportunity for understanding genetic variations, gene expression, epigenetic modification, and microbial diversity. So NGS has been instrumental in identifying disease-causing variants, uncovering novel drug targets, and shedding light of complex biological phenomena, including the heterogeneity of tumor and developmental processes. So, next generation sequencing has transformed the scientific landscape, expanding the scope of inquiries in various applications within the medical field. Notably, applications include whole genome sequencing. So, it is repeat sequencing of entire genomics, providing comprehensive genetic informations. Targeted sequencing, like deep sequencing of specific target regions for detailed analysis. RNA sequencing, discovery of novel RNA variants and splice sites, and quantification of mRNA for gene expression analysis. Epigenetic analysis, like examinations of epigenetic factor like genome-wide DNA methylation and study of the DNA protein interactions and nucleus gene regulations. Cancer genomics, like sequencing of cancer samples to explore rare somatic variant and tumors of clones, and comprehensive analysis of cancer genomics for a better understanding of targeted therapeutic. Human microbiome studies, like investigation of the microbial communities residing into the human bodies and understanding the role of microbiota in health and diseases. Pathogen identifications, like pathogens, detection of novel pathogens for effective disease management and preventions. So the versatility of NGS, coupled with advanced sample preparations and data analysis method, empowering laboratories to address diverse research questions. This application showcases the technology's potential to unravel the genetic complexities, contributing to advancement in medical research, diagnosis, and personalized medicines. So once again, thank you, Mr. Sohni Tagore, for gracing us with your presence today. We greatly appreciated your time and knowledge. Now, audience, before we begin to this our webinar and introduce our esteemed guest, allow me to provide a brief overview of Heredity Biosciences. Heredity is established in 2018 and rebranded in 2021 as a leading provider of research and training services for hospitals, life sciences institute, and pharmaceutical firm. It is a trusted name of medical and scientific community. We boost a sessions team specialized in genetic, proteomics, metabolomics, bioinformatics, scientific communities. We are committed to the quality, aim to the frontline technologies. Our collaboration with the Heric Life Sciences Private Limited with impactful innovations to collaborations in life sciences and healthcare research globally. So Heredity Biosciences is a proudly operating for the two locations. As you know that our main office is available for this Mehta Lagoon Road, Bhubaneswar, and we are already already new ones opened in Guwahati, Assam, inaugurated last year. This expansion remark as a substantial milestones reflecting progress and growth. It enhances our capability, fostering collaborations with local institutions, so that we are widely expressed to this research as well as for this services part. We anticipate a meaningful contributions to the advancing life sciences through the exciting the growth. So, Heredity Biosciences excel to the life sciences and healthcare, offering the comprehensive research and training services. Our life sciences services, as you know, that we are providing with genomics, proteomics, and metabolomics, and it is not only for the Odisha. We are outside for the outside. It accessible the national wides. In healthcare research services, we investigate to the crucial area of research like clinical microbiology, infection genetics, cancer genetics, fertility research genetics, and conventional qPCR analysis. Explore the wide details available in our databases websites also. So, Heredity Bio Academy is a Odisha's leading training institute. We are proudly offer comprehensive hands-on training research, training of the student and the professionals. These are listed course modules provided comprehensive hands-on procedures. So some specialized modules already introduced for that. So every like clinical embryology, that is a male infertility part, histochemistry, immunohistochemistry, qPCR, genetic diagnosis. This is the frontline modules are designed to address to the growing need for advancement in the field of biosciences or life sciences professionals and the students. More information we already have in the websites. Heredity Biosciences excited to announce dissertations program. Personalized for the life sciences students. This program offers unparalleled to the hands-on exposure to advanced experimental laboratory practices, providing the holistic learning to experience in the field. So the students, if you are enrolled to this program with the research in a frontline experimental technologies, gaining individual practical skills that complete academic learning. So the importance of the development of the deep understanding and advancement of laboratory practices within the life sciences domain. So Heredity Biosciences is committed to all the students and providing for the dynamic and enriching environment that ranging for the next generation's life sciences research as well as for the hands-on practices. The dissertation program stand to be gateway to acquiring essential skills and expertise, preparing to the students to real-time challenge and opportunity to the landscape of the biosciences. As you know that we have good infrastructure, world-class facility, centralized air conditioned laboratory facility, good Wi-Fi environment, complete research learning facilities, well-trained and experienced trainer and demonstrator, complete hands-on individual experimental exposures to one-to-one exposure, publication assistant and placement assistance also to all the students. So, in the past 3 to 6 months, Heredity Biosciences has reached to the milestones to witness the successful spread to the placement activities. This is the placement activities of the 5 to 6 talented students are joined for the different sectors, like for different fertility sectors, overseas, and Bila fertility and other fertility, and as well as for this industrial sectors also. So we are extend to heartfelt Congratulations to all these exceptional students to wish them to the journey filled with the prosperity and fulfill their beginning of the career. May their contribution to the field of biosciences and the healthcare is impactful to your your for the future career. So cheers to the success, to the promising, to the path ahead. So, as you know that we have research and publication services. Every month we are trying to publish to these research activities. So we have more than of these publications we have, and more than 2 to 3 patents right now are already patented. Hereditary Biosciences celebration for this the second Foundations Day that is promised to make to the our inceptions. The festival at our office we filled with laughter, friendship, and sad to the moment of the joy, and memorable and fruitful events to 8 of December. So thanks to everyone who contributed to this, and cheer to more milestone ahead to joining to these events. So we have a newer celebrations to our internal our teammates, and we are celebrating a lot, and we will enjoy a lot for the celebration of this new year. Now, ladies and gentlemen, it is with great pleasure that I introduce our esteemed guest speaker for today's webinar, Ms. Sohni Tagore, currently serving as a research analyst at the Genebox in Mumbai, Maharashtra. Ms. Sohni, academic journey, stand as a testament to her unwavering dedication in the field of medical genetics. Starting with her academic pursuit, Ms. Sohni completed a BSc in medical genetics and genomics from the University of Glasgow in 2018 to 2019. Her groundbreaking work included in the depth analysis of RNA sequencing data from uh, global customer stem cells, aiming to identify potential therapeutic targets. She holds a BTech in biotechnology 2013 to 2017 from DY Partial University School of Biotechnology and Bioinformatics, and PG Diploma in Physiological Counseling and Physiology in 2016 to 2017 from Indian Institute of Counseling. Ms. Soni's Rees experience including beginning a research student at MGM New Mumbai Hospital, Vansi, Mumbai in 2016 and 2017, where she worked in the field of cytogenetic and genetic counseling in the genetic department, focusing on children affected by the Down syndromes, cerebral and ASD. So here journey continued as a research intern at the PIM Biotech for 3 months, where she contributed to an international educational internships and result recruitment for the company of a pilot project, enhancing awareness about NGS data analysis and genomics. In 2019, she served as a research student at the University of Glasgow for 4 months, concentrating on the analysis of RNA sequence from the glioblastoma stem cells. Her proficiency include work with the software such as DAVID, cytoscopes, Panther, KEZZ softwares, ST, STRING strings, strict softwares. From October 2020 to December 2021, Ms. Sohni was a researcher at the University of Antwerp, leading to the project in the expansion of diagnosis, yield of genetic testing, and continuation hearing loss. Presently, she is contributing as a research analyst at the Genebox, Mumbai, Maharashtra, showcasing her skills in genomic analysis and natural language processing with strong research backgrounds of 3 years in clinical genetics. Ms. Sohni's certification include autism spectrum disorder, NGS data analysis, and Linux basis epidemiology, the basic science of public health, and STEM ambassador. She has been honored with the Scotland India Scholarship, and she published on the genetic study in children with Down syndrome and autism like behavioral diseases. Today, Ms. Soni will share her invaluable thoughts in a webinar titles Medical Genetics and the Advancement in Next Generation Sequencing. We eagerly anticipate the wealth of knowledge she will impact. Thank you, Mr. Swani, for joining us, and enthusiastically look forward to an enlightening and enriching session ahead.
Thank
This is the team, Heredity Biosciences. Team Bhubaneswar and Team Guwahati. They are contributing a lot for this science and scientific research. Thank you.
Thank you so much, Dr. Gopal, sir, for your kind words and the great introduction of me. So I think we can start with the presentation. Hello, everyone.
Yeah, to start with.
Okay, I'll just share my screen. Is it visible?
Ma'am.
Okay, great. So I'll start with that. So as you all know, today's topic is medical genetics and the evolution of NGS. So some of the topics that I aim to cover in today's presentation are introduction to medical genetics. Why is medical genetics important in today's age? The general overview of next generation sequencing technologies, advancements in the field, and the scope of NGS and medical genetics in India and worldwide. So as we all know, genetics is the study of genetic variation, genes, and heredity in organisms. Genomics encompasses the whole study or a part of the genetic and epigenetic sequence information of organisms, and it attempts to understand the structure and function of these sequences that we have in our body. So all of the DNA information that we have stored in each of our cells, that is in totality considered with genomics, and it can be done for any and every organism on this planet. So medical genetics is basically the application of these genetic principles that we study in medical practice. And this includes the studies of inheritance, mapping disease genes, diagnosis and treatment options, and also genetic counseling, which has become very important in today's time. And another field that we are currently focusing on a lot in medical genetics, which is a subfield of it, is pharmacogenetics. That is the study of how drugs affect the body with respect to specific genetic backgrounds, and the knowledge of these drugs and how it can affect and improve effectiveness of drugs in our body and minimize the side effects on an individual patient basis is what we call personalized medication. And it is a part of pharmacogenetics. And in a broader umbrella term, it is a part of medical genetics. So why do we need to study genomics? It gives us innovative ways to diagnose a disease, and early detection of any genetic disorder helps in identifying a genetic predisposition to a particular disease like type 2 diabetes or obesity or anything else that you might have in the future. And it also gives us ways to have new approaches of treatment that can be utilized by pharmacogenetics. And that is why it is considered in combination with medical genetics. And this development of drugs helps in targeting specific mutations like how we see in cancer genetics. So in broader terms, there are 4 types of laboratory genetic practices that we encounter. So first one is molecular genetics. So molecular genetics is basically, it applies to the investigative approach to determine the structure and function of the genes in an organism's genome. And cytogenetics, which I worked with during my bachelor's dissertation at MGM Hospital, Vashi, was the study of the structure and properties of chromosomes and their behavior during somatic cell division during growth, that is mitosis, as you all know that term, and also germ cell division during reproduction, which is meiosis, as well as their influence on the specific phenotype leading to disorders like Turner syndrome or Down syndrome. And then the next one is molecular hematology. This is the study of blood and its production from molecular and a genetic perspective. So you might have heard of the terms like sickle cell anemia and thalassemia. So these all come under hemoglobinopathies, which is the study of abnormalities in the globin chains of hemoglobin, which is our blood molecules. And in addition to this is also known as a broader umbrella term of hemoglobinopathies, which entails other diseases which come under molecular hematology. And then the next one is molecular pathology, which is the study of disease at the molecular level. So this includes molecules found in tissues or organs or even bodily fluids like urine and stool. So these all are 4 of the components which make up laboratory genetics. So you can go into any of these fields depending on your liking. So then now during the testing of genetic testing, what all tests do we have? So the first one is a diagnostic test, which is basically to diagnose diseases that we have. It can be either genetic or otherwise also, like the blood test that we do. The next one is carrier testing. So if a family has a child affected by a genetic disorder, how will they know their chance of recurrence for another child carrying the same genetic disorder? So there is a chance that the mother or father is a carrier. So the carrier testing is very important in that regard. Then disease monitoring. So if you have any disease, to develop a drug for that. Predictive testing is basically to predict what are the chances that you might have of developing a disease in future depending on your habits. So what further steps you can take to abort those diseases in good time so that you don't get affected by them. So that is predictive testing. Now, pre-implantation is basically a genetic diagnosis service in conjunction with the assisted conception unit. So that is mostly with the fertility clinics that we have. And it is a part of clinical genetics. And it is also offered for as a biopsy testing using fish for translocation carriers and pregnancies at the risk of X-linked disorders. And prenatally, the last type of testing is where we offer karyotyping and QF-PCR and test using amniotic fluid and CVS. And in together, all of this, for cancer referrals, we offer diagnosis and disease monitoring by further techniques that will come up in the future slides. And this helps in making informed decisions regarding treatment, particularly where targeted therapy is available, like in cancer, where we have tyrosine kinase inhibitors. So this is all a part of testing that we have in genetics, medical genetics. So now, during cancer, we have something called as somatic testing. And somatic testing is usually done, just a second, I think I missed a slide. Yeah, somatic testing. So somatic testing is usually done after a person has been diagnosed with cancer, and it looks for genetic mutations in tumor or cancer cells. So it is very important to understand the hematological malignancies and the solid tumors and how they affect a person's body when they have been detected with cancer. And also these techniques help in the detection of minimal residual disease, which is basically a term used to describe the small number of cancer cells in the body after a cancer treatment has been done. So after you've undergone chemotherapy and radiotherapy to eradicate the cancer cells in your body, there still might be some cancer cells left. And this is detected by the use of MRD, or minimal residual disease technique. So in this, you use a small sample of your bone marrow or blood. The doctor extracts it, and the doctor can then determine how many cancer cells remain in your body. So the MRD testing is so sensitive that it can find one cancer cell in 10,000 or 1 million healthy white blood cells also. So it is very, very specific and sensitive to the cancer cells in your body. And how this will help is, it will help you to classify that malignant disease and then target the therapy for the specific drug treatment options with it. So next is germline testing. So germline testing is if a genomic tumor testing has happened and it identifies a pathogenic variant in a gene associated with a hereditary cancer, suppose. So someone in your family has a cancer and you are scared that what is the chance that you also might have that. So germline testing gets very important in this regard, because then you have to get the genetically targeted treatment, and also it helps you identify relatives and other family members who might benefit from the personalized cancer screening and prevention. So it allows confirmation and exclusion of diagnosis for hereditary disorders as well. And germline testing is also important, apart from cancer, in terms of carrier testing and risk assessment in families with a known genetic disorder. And so the predictive testing, as I had explained in the previous slide, is also how it comes into play, because individuals at risk of a late onset hereditary genetic disorder are also benefited from germline testing. And prenatal diagnosis as well is a part of germline testing. And it is a case-by-case basis. So depending on the clinic and the doctor, how he sees the further, the actions to be taken, they will allot you which test is the best for you. So some of the techniques that come under the umbrella of medical genetics are firstly karyotype. And this has been like the process for the longest time before even sequencing happened. So karyotyping is the process of pairing and ordering all of the chromosomes of an organism, and then putting it beneath a dye, and then you observe it under a microscope, and then you order all of the chromosomes of the organism, and then you see it as a genome-wide snapshot of your individual chromosomes. And in line with this is FISH, which is fluorescence in situ hybridization. And it is a molecular cytogenetic technique that allows, the localization of a specific DNA sequence or an entire chromosome in a cell. The next one, array or a microarray, is a laboratory tool used to detect the expression of thousands of genes at the same time. The next one is MLPA or MALPA, which is the full, in short, it is MALPA, but the full form is multiplex ligation dependent probe amplification. And this uses pairs of probes to detect specific sections of the genome. And it is mainly used to identify exon level copy number variation. And the next one is PCR. We are very familiar with PCR. That is like RT-PCR during COVID was the rage. So sabko pata hai ki PCR hota kya hai. PCR basically involves using short synthetic DNA fragments called primers to select a segment of the genome to be amplified, and then multiply rounds of DNA synthesis to amplify that segment, and then we observe that. The next is sequencing, which is Sanger sequencing. And the next one, the last one, not the least, is NGS. And we are going towards better technology. So I'll explain about sequencing and NGS in the coming slides. So the sequencing technologies, firstly, Sanger sequencing, or the first generation sequencing, by which we did the human genome project, which we completed in 2003 after like years and years, 20 years of hard work, was the most useful and effective in sequencing DNA fragments of 500 to 900 base pairs. And it is widely used to sequence organisms that have short DNA sequences or DNA fragments that we amplify by PCR. Then NGS is our second generation of sequencing, which has become very popular in the past 10 years. It is also called as massively parallel sequencing or high throughput sequencing. So sequences like short DNA fragments, typically ranging from 50 to 300 nucleotides in length, are used for next generation sequencing. And currently we are on the cusp of going into third generation sequencing or long read sequencing. And the basic principle behind this is to measure changes in the electrical properties of DNA as it moves through the nanopore membranes or channels. And when the fragment crosses the channel, the platform identifies the exact nucleotide base and gives you the sequence of the DNA in real time. We'll look at this in the further slides. So now, NGS types. So for the whole second generation sequencing, which has become very popular in these last years, we have 3 types, which is first targeted sequencing, where we have a selection or a capture of a certain region of the DNA and the genome. And we have a specific panel of genes of interest, typically 200 to 20 to 200 predefined genes. And they are then sequenced in great depth for them to be more cost effective. So if you know that you have a particular phenotype of a disease, then you go for targeted sequencing. Next is whole exome sequencing, wherein we take all of the exons of all the known genes, which is basically 1.5 to 2% of all the human DNA. 98% of our DNA is junk, as we all know. So this whole exome is taken, and then the variable read depth at the boundaries of the exons are also considered, and this gives us a good sequencing depth, and it is more cost effective than whole genome sequencing. Whole genome sequencing is basically you take the entire human genome and you analyze it, including the RNA, the introns, all of the junk DNA that we consider. This gives you a moderate read depth of 10 to 20x. That is the magnification you can achieve. And it gives you a similar read depth throughout. And it may also detect copy number variants and repeat experiments. But this comes at a very high cost. Now, between whole exome sequencing and whole genome sequencing and targeted sequencing, there is another area, which is clinical exome sequencing, or CES. Now, this is specifically used by laboratories for identifying disease-causing DNA variants within the 1% of the genome. So whole exome identifies like 1.5 to 2%. And targeted sequencing is 20 to 200 genes. So clinical exome is somewhere between that, and it takes only the 1% of the genome, which codes for the proteins and exons and the splice junctions, and it will examine that. And in the absence of a well-defined clinical presentation of a disease, WES enables the scanning of the entire coding region of the genome at a reasonable cost, while clinical exome sequencing is a subset of WES, and it typically targets 3,000 to 6,000 genes. So it is more a little cost effective than whole exome sequencing also. So now, what is the parameter and how do we go about doing whole exome sequencing? So the first part is you take your double-stranded DNA, wherein you have the exons and the introns, you do the adapter ligation in your mix, and then you hybridize it to a array for further target enrichment of the whole exome sequencing, and then the computer generates the sequencing output. Now, this is the laboratory preparation part. What happens when you do put it into the computational bioinformatic pipeline is that it will do a base calling of all of the data that has come out from the adaptation target enrichment site, and then you can have a read data where it will align. You will use a program to align it to the human genome and then align the reads to it and then perform read trimming. And this will you will use another program to do basic statistic analysis on the exonic reads, and this will give you an output data of SNVs, that is structural variants, and indels, which is insertions and deletions. And then you can filter and annotate this data to get disease-associated novel variants. So the data of sequencing is very confusing and unreadable for most of the bioinformatic people. So to make this sequencing easier, analysis easier, we use short fragments of DNA, which require sequence assembly to figure out the final result. And this is why the computational pipeline is used by the researchers who are doing whole exome sequencing, so that they can do an easy analysis by variant calling for the genetic diseases. So next is whole genome sequencing. So as you can see on the picture, this is like the best diagram to explain whole genome sequencing. In the reference genome on the left-hand side that you see, there is you break this genome, like by enzymes and other laboratory prep practices. You break it into large fragments and clone the genome, DNA. And then you break it into individual clone smaller fragments, and then you sequence the thousands of reads that are generated from it and assemble the sequence reads for each clone. Now, this is done differently for the reference genome, and then you have the individual genome. So here you do the same laboratory practices, but you break the genome into smaller fragments than the reference genome. And then instead of the thousands of genomes that were collected by the reference genome, in the individual genome, because you have smaller fragments, you will get millions of sequence reads, and then you align these sequence reads into the reference genome, which you already have. So this will give you the individual genome of that person. So how does the output data come in? It's firstly, you filter out the variants based on frequency in databases. And this is like variants with damaging effects like high protein function scores, in silico analysis, which is done by SIFT, CAD, mutation taster. All of these softwares are used. In silico is bioinformatic computer work, jo hum karte hain, uska. Then you use conservation scores to rule out most of the variants which are damaging effects. So you make a smaller data out of the big chunk of data that you have. And conservation scores are basically philo-P, if you are familiar with that. So the next is, once you have the highly damaging variants with you, you select candidate variants by segregation in the family, if you see any segregating data or anything like that, and you use it for Sanger sequencing. And you use a control group and the candidate gene variant that you have to see which is how the variant, which is like the candidate gene that you got from your in silico analysis, affects with the control group. And the next is, once you have successfully found that, okay, this gene is damaging or this is the variant which is causing the disease, then you can't just say ki yahi disease cause kar raha hai. You have to do a functional validation and gene characterization of that variant. And this is done by in vivo, which is mouse models or any other animal models that we have, and in vitro models, which is cells and organoids, spheroids, जो भी हमारे आजकल हम उसके लिए सबसे बेस्ट है, वो यूज करके सेल लाइन हम उसका फंक्शनल वैलिडेशन करेंगे. So next is, I'll walk you through some examples of how do we do the screening via NGS. So the first is, you have a disorder which has a genetic basis, but the phenotype, so you can't really see clearly कि बहुत सारे symptoms हैं. The child or the person has a lot of phenotypic symptoms, but there is no one symptom which is standing out to give you a diagnosis that this is it. So the patient may have seizures or encephalopathy. So what do you do in this case? You sequence all the genes by whole exome sequencing or whole genome sequencing. Next is, the phenotype is present and it suggests that there is a genetic disorder. So for example, maybe the patient has long QT syndrome is what you're thinking from the phenotype. So what will you do? You will perform targeted gene panel testing to establish a diagnosis. Next is, there is a single gene, you know that it's a cancer, and this is like a Li-Fraumoni syndrome wala cancer, or there is a biotinase deficiency. So the phenotype is very clear. So you will do Sanger sequencing, and that will confirm your diagnosis that this is what is the person affected by. Now next is long read sequencing, or the third generation sequencing that I was talking about. So long read sequencing is basically complex genomic regions of our DNA that we have, which may contain highly repetitive sequences, which can also have duplications of the genes or pseudogenic sequences, which complicate the analysis and mutation detection. So the current DNA technology, sequencing technologies that we have, which is NGS and Sanger sequencing, are often not able to unravel these regions or provide insight into phasing. So what is phasing? Phasing is the process of assigning the alleles A, T, G, C जो हमारे DNA में होते हैं basis to the paternal and the maternal chromosomes. So this will provide help in phasing or the exact location of the potential pathogenic variants. And currently, the two most used technologies for long-read sequencing are hi-fi reads from PacBio and Oxford Nanopore sequencing. And Oxford Nanopore sequencing with Minion and Flongle was the technology that I was using for my project in Belgium in hearing loss. So here next is a short video of how the nanopore sequencing works. So this is the flow cell wherein you put in your DNA sample and there is a motor protein and adapter sequence. Now here you see these channels are the nanopores, and this has 512 channels with 4 nanopores in each. So that is 2048 nanopores. So once the tethering happens, it will be supported by microscaffolds, and the electronic sensor chip then unwinds the DNA. So from double strand, it will make it single stranded. And then each channel, which is associated with the specific electrode in the sensor chip, which is controlled by the nanopore, is measured individually. And this will generate an ionic current, which is passing through the nanopore with a constant voltage. And this is applied across the whole 2048 membranes that we have. And because the trans side is positively charged, it will release the DNA molecule into a single strand, and then you can see the ATGC sequencing in real time on your computer screens. I hope that was helpful. So the next is, with long read sequencing, there also comes the problem of splice site mutations and how do we test that. So a splice site mutation is basically a genetic alteration in the DNA sequence that occurs at the boundary of an exon or an intron. So that is called a splice site. As you can see in the figure, there is exon 14 and there's exon 15. And then there's this yellow part where there is a 97 base pair intron insertion, retention. So wo intron nikla hi nahi. So you have a splice site therein. So this change can disrupt the RNA splicing, resulting in the loss of exon or the inclusion of intron. As you can see in this, there is an intron retention, and this will result in an altered protein coding sequence. So the splice site variant, what happens is, for the precise mRNA splicing, it is essential that there is appropriate protein translation, which has not happened in the previous case. So there will be defective splicing, and this has been increasingly recognized as a disease-causing element. So whole genome sequencing allows us to systematically detect non-coding variations and identify pathogenic deep variant, intronic variants that lie more than 100 base pairs away from the exon-intron boundaries. So the ideal method to determine how this variant affects splicing is to perform reverse transcriptase PCR or RT-PCR. Again, we know it from COVID, RT-PCR. And this will show how the transcripts are affected by the variant using extracted RNA from the tissue of the patient. And there is a necessity of the analysis of the regions outside of the coding exons, especially in families with multiple affected people, and evidence of linkage to the known disease loci. And this development of new computational tools based on deep learning network like Splice AI, it's a computational tool which has greatly improved our ability to predict cryptic splice site variants. But there is still a need to supplement these bioinformatic predictions with in vitro functional assays to identify and validate deep intronic splicing variants. So now coming back to genetics, enough about NGS and long read sequencing. What happens in the genetics clinic? So firstly, we have the prenatal genetics, wherein we look at couples who are at risk of having a child with a genetic disorder because of preconception or while they are pregnant. So high risk prenatal screening results, if they did a genetic testing or they did an ultrasound and they got some result which points to the child having Down syndrome or any other karyotype anomaly. So that is what is seen in prenatal genetics. And then there is pediatric genetics, which focuses on birth defects, developmental disabilities, autism, epilepsy. So this was all a part of my uh, experience at the MGM New Bombay Hospital, Vashi, wherein I was dealing with pediatric genetics and children with Down syndrome, cerebral palsy, and autism spectrum disorders. So I had in total analyzed 16 cases for that. Then the next is cancer genetics, wherein you can have any type of cancer, but I'm just giving some examples like breast, ovarian, bowel, or cancer or endocrine tumors. And then there is adult genetics, wherein you have any of the cardiomyopathies or heart-related dysfunctions or arrhythmias, inherited kidney diseases, dementia and neurodegeneration, Alzheimer's, any of these connective tissue disorders also are included in adult genetics. Oh, sorry. Yeah. So why do we need genetic counseling? So genetic counseling basically is to counsel people who are planning to get pregnant, or for people who have a family or a relative, someone who is affected by a particular genetic disorder. So this enables the families to understand why genetic testing was done, and then also lessen the burden of guilt that they face, and also provide treatment options if any are available. And then also address the at-risk family members if any are present for that same disease. So suppose if someone's aunt has breast cancer, the chances that if she has a sister, she also might be affected by some form of cancer is really high. So this will help address the at-risk family member, which is the other aunt, for that cancer screening. So it will help you give treatment options to them, and also provide accurate recurrence risk for future pregnancies for any couple who has a child affected with a disorder, or they know that we might want to plan another pregnancy, but we don't want the child to have that particular disorder that the previous child already has. So it can help you give a recurrence risk for the future pregnancies, and then the couple can make an informed decision whether they want to carry forward the pregnancy or not. So the scope of the field. Currently, there is a lot of buzz going on around NGS and medical genetics. There is a hype, and medical genetics basically encompasses all of the many different areas that we have in the clinical practice. So there is a collaboration of physicians, genetic counselors, nutritionists, clinical diagnostic laboratories, the geneticists, the bioinformaticians, and everyone who come together to give a particular risk and research into the causes of inheritance of genetic disorders. And the examples of conditions that fall within the scope of medical genetics is huge, and it includes birth defects, dysmorphology, intellectual disabilities, autism, mitochondrial disorders, skeletal dysplasia, connective tissue disorders, cancer genetics, prenatal diagnosis, everything. So medical genetics is increasingly becoming very relevant to many common diseases, and it is being used for endocrine disorders also, cardiovascular, pulmonary, ophthalmologically, the eye disorders, the renal disorders, psychiatric and dermatologic conditions as well. So the medical genetics community is heavily involved with individuals who have undertaken elective genetic and genomic testing. So the people who know that they have some genetic disorder, the physicians or doctors in today's time, they refer them to geneticists so that they can have a proper guidance and diagnosis as well, and also to make an informed decision. So the current scenario in India is basically we have all of the latest technologies available. We have NGS. Most of the high-end labs are working with NGS tools. And we are offering very competitive costs because our population risk is load is huge, right? We are a country of 1.4 billion people. So we have to decrease the costs. Otherwise, it is very hard to bring it into the mainstream healthcare. But the other issue that happens is it is still very expensive and it is not very affordable to the common man. And also there is no insurance coverage provided in India. So it will, in the long term, I hope there is an early molecular diagnosis which will help improve the patient outcomes, and the healthcare expenditure is also curtailed, and the government makes some strides toward making these technologies even more viable for the common man to be available. So thank you so much, and I hope you all liked this session. And I would be happy to take in any requests. If you have any further sessions that you might want with me, you can reach out to me via my contact details on WhatsApp. You have my number. You have my email, and via LinkedIn as well. If the profile is Sohni Tagore, please feel free to reach out and talk to me and let me know if you want to have further sessions going in depth about certain things. And please spread the word if you liked it. Thank you so much. Hi, sir. Sorry, I can't hear you. I think you are on mute, Dr. Purohit.
Now it's audible.
Yes, sir.
Excellent webinar today, and
Thank you
it's
so much.
wonderful. But I request to each and everyone to the audience, so at least one or two questions. So
I think they're typing in the chat with the questions.
someone is no, something is
Excuse me. Hello. Hello.
Hello.
Ma'am, is it audible?
Yeah, I hear you, Mr. Senthil. Yeah.
Yeah, ma'am. Ma'am, my doubt is about how can we pursue our career in the medical genetics field, like
Okay.
which way?
So if you have a like a degree in biotech or biological sciences, there is this MSc in medical genetics at the University of Manipal, I think, if you want to stay in India and do it. So they have their session start in September, I think, and they have their entrance exams during the month of May or June. So you can just look at the website of University of Manipal, go and check it for MSc Medical Genetics, and you'll see how you can go in there.
Yes, ma'am. Thank you. It's clear.
Yeah.
So one question from my side.
Yes, sir.
So how does germline testing differ from the somatic testing in
So,
identifying hereditary disease? How does
yeah,
germline testing
germline testing. Yeah.
differ from the somatic testings in the identifying hereditary diseases?
How does it differ? So basically, as far as I know, it is germline testing is for all, like the autosomal cells, not the autosomal, sorry, the germline cells in your body. And somatic testing is for just the cancer cells mostly, because once the cancer has been detected, it will be present in mostly the organ which has been affected. So that is the somatic or the other cells. And the germline will be for the hereditary diseases. So the testing, if you go by the, are you asking for the parameters or the different genetic tests that happen?
Only test. What type of test? So,
I'll have to get back to you on that. I'm not very sure right now, but I'll get back to you on that. Yeah,
so
yeah.
what are the challenges exist in integrative NGS into routine clinical practice for the
The
disease diagnosis perspective?
integration of, sorry, your voice is breaking.
What are the challenges exist in NGS into a routine clinical practices?
NGS in the challenges for NGS in routine clinical practices is the question, right? Okay. Yeah. So the challenges are basically the cost. It is still not very affordable. Even though we say that the cost has come down, it is not that low for the common man to be affordable. And also there is no awareness. And
Yes.
when we go for the technical testing, it is, there is still not a very high awareness amongst doctors also, that how much medical genetics or these NGS technologies can help you with the diagnosis. We are still very much on the like सारे टेस्ट करेंगे हम लोग, पहले ब्लड टेस्ट करेंगे, ये वाला टेस्ट करेंगे, MRI करेंगे, जो भी करेंगे, एंड देन हम लोग जाके कुछ नहीं मिला तो फिर हम जाके सोचेंगे कि लास्ट में चलो अभी जेनेटिक टेस्टिंग करवा के देखते हैं, शायद इनका कोई जेनेटिक कंपोनेंट हो. Otherwise it should be like in first world countries, like it is happening, the first time you see a phenotype, you should be going for a particular genetic test. And we have targeted gene panels of multiple diseases which are already present with the predetermined genes in it. So it is still not happening. So I think the main challenge is basically awareness. And we need a good team of genetic counselors also to counsel those patients after the test results come out. So it is very important for them to, feel okay with the test result and be like, ha, theek hai, we can, there is a chance that we can live with it, or, the emotional guilt that you feel, that has to be taken care of by a doctor. And our doctors, unfortunately, they are so burdened with a lot of patients, they don't have time to give personalized attention to such cases. So that I think is very important.
Okay. So audience side, any questions? So what does the NGS play in
Hello,
parental
ma'am.
diagnosis? Yes, Nikita Panda.
Ma'am, I just want to ask that as you said that if a mother having cancer, then the sister also has the risk to having the cancer.
Yeah, there is a
So,
risk. There's always a risk.
so,
Yeah.
so, ma'am, can we consider the cancer as a genetic disorder or hereditary disorder?
No. So this is how it gets tricky. Cancer, in actuality, 90% of the cases are sporadic, and only 10% of the cancer cases are hereditary. This is a published paper by which I'm saying these numbers. I'm not making these up. But so it's not always that you will get cancer if someone in your family has cancer. But if someone in your family has cancer, and if they are like of your immediate family, the risk becomes higher. And then it is the doctor's duty to inform the patients that the at-risk family members that there is a chance that this might happen, so you might as well go for a predictive screening or a cancer test every 4 or 5 years if they are like 40 years or above, or 3 or 4 years kid gap may if they are 50 years or above, to mitigate that risk. So there is a chance, but it's not that कि अगर किसी एक को है तो होगा ही होगा बाकी सबको, ऐसा नहीं होता. Did that answer your question, Nikita?
Yes, ma'am. Thank you.
Okay. I think I see a question in the chat. When any lady was pregnant, then at the time of 4 months of conceiving, doctor advised to go for genetic analysis of infants and took charges of 10,000 to 15,000 for the same. So would you please be able to tell me, is it possible to test all the genetic testing within that price, or just a formality to go for the test? I can't really answer the question for the prices that are being charged, because it's very different and it's on a case-to-case basis. So I can't really say on the cost or why they would charge for that particular test as well. No, I can't really comment on this. Sorry.
Okay. So any questions from others, audience sides?
Hello, ma'am. Good evening.
Good evening.
Ma'am, I was asking that I have done my master's in zoology, and if
Okay.
I pursue my career in PhD in medical science, medical genetics, then what will be the opportunity after that?
It depends on how you like. PhD is a very specific topic, right? So what will you do your PhD on will matter a lot. So also your supervisor and the whole journey you go through with the PhD will determine the outcome and what career prospects you will have after your PhD. So that will depend on you totally, where you want to take that direction.
Okay, ma'am. Thank you.
Yeah.
Okay, thank you everyone. So
Okay.
thank you for this great sessions today, and finally we will be end of the sessions. So hope everyone should be enjoyed to this webinar, and so finally on the behalf of Heredity Biosciences, we will close to this sessions also. On the behalf of Heredity Biosciences, I extend my heartfelt gratitude to each and everyone to be being a part of this insightful webinar today. This webinar is truly been a remarkable journey into this webinar topic, that is medical genetics and the advancement in next generation sequencing. So thanks to Ms. Sohni, your expertise and knowledge in the field of medical genomics has a truly commendable, and we are really honored to have this opportunity to learn and many disease things and many things we learn to this. As for the audience side, I really thankful to you, and your presentation has been enlightened, offering really valuable insight into the significant parent tribes diagnosis and the role of advanced research into this diagnosis. I would like to acknowledge dedicated effect all these members to patients to join to this end of this sessions, and also thankful to this huge months you are accepting our invitations, and so you are given for the valuable times. So it is 8 o'clock, 8:30, and you are given for the times. So it's really honored on behalf of Heredity Biosciences, and I'm really heartfelt thankful to you. And for the future, we will try for that.
Thank you so
To
much.
coordinate any contributions and any things about yourself.
Yeah,
So,
sure.
I would like to acknowledge to dedicated effort to our team members also. Sushree.
Yeah, thank you so much for reaching out and coordinating the event so beautifully. Thank you,
We are
Dr. Purohit.
coordinating from a long times to coordinate you. So I hope you are. They are communications to all these valuable thought, whatever that we are required. You are required for that, and so heredity is only for the organizing for the um, for our for the sharing to the thought to these exports. So we are only for the empowering to the students or professionals to that they should know that what is going on to these markets, what is actually things into this market. So they should be utilized to the career to this to further future career. Once again, thank you, Ms. Sohani, for sharing your expertise with us, and thank you all for the participation and make the webinar resourcing success. And we look forward to hosting more enriching event in this future. Thank you.
Thank you so much. And all of you, please feel free to reach out to me and let me know if you want more sessions. Thank you so much.
I will say to your mail ID also, if anyone to ask any questions, it will be said to.
Please share my contact details from my last slide. If anyone wants to come back to me with more questions, more requests, I'm open. Thank you so much.
Thank you.
Thank you so much for providing me with this opportunity. Thank you so much, sir.
Thank you.
Thank you. Good night.